Can I stop? Okay. Bismillahirrahmanirrahim. Good evening, ladies and gentlemen. This session, we are inviting Dr. Muhammad Salim, Associate Professor, Department of Chemistry, Ghazni University, Darul Khan. And he has a certified research publication and about 60 highly cost doctor again from South Africa.
Bismillahirrahmanirrahim. Very good evening, dear students. Assalamu alaikum. Thanks for introduction. Today, the topic of my talk is multi-component synthesis of 2,4,6 tri-pyridine analogs as antibacterial and antiviral. Actually, we have used some novel routes to synthesize the pyridine derivatives. We have not used the pyridine. In this research, 5 novel pyridine derivatives have been synthesized under the umbrella of green chemistry. The synthesized derivatives were evaluated by quantum spectroscopy and proton and mass spectrochemical technique. These compounds were assessed for various biological activities such as antibacterial, antioxidant, and urate inhibition. These compounds showed excellent results for urate inhibition and antibacterial activities. What is a multi-component synthesis? Actually, this is various fragments when combined together and form sometimes very simple and sometimes very complex molecules. This is called multi-component synthesis. Actually, pyridine is a fundamental anhydrocyclic molecule. It is discovered by the Anderson in 1846 and its structure was constructed by Wilhelm Conner and James Stewart. It has a planetary and conjugated system of 6 pi electrons. Pyridine molecule is essential of alkaloids, nicotines, and vitamins. The natural resources of pyridines are vitamin B6 and paradoxal and some nicotine. These are the natural resources of pyridine. Pyridine analogs are dominant in the cell system, involved in enzymatic activity. They acted as convergent nucleotide adenosine diphosphate that participated in different addition and substitution reactions in the cell. You can see in slides that pyridines play an important role in the catalytic activity of some reaction in the cell. Importance of pyridine analogs. Pyridine molecules possess diverse structural features. Pyridine analogs present numerous biological activities such as antibacterial, antiviral, anti-malarial, anti-convulsant, anti-hypertensive, antipsychotic, anti-cancer, and anti-inflammatory activities. That's established multiple role of pyridine molecules in pharmaceutical and chemical industries. Synthetic applications, many synthesized pyridine compounds play a prominent role in drug industries. As an example, as soon as it is anti-tuberculosis and sulfapyridine as antibacterial agent. Here is the structure of isonazole and sulfapyridine. Here, pyridine is the major precursor which is used as anti-tuberculosis agent. Pyridine varieties also achieve a significant importance in herbicide pocket and fungicide. That is the structure in which pyridine is the major role. Furthermore, pyridine derivatives are efficient in cure of Parkinson, hypoxia, asthma, cancer, and kidney problem. You can see slides that some pyridine analogs play a major role for the treatment of very complex diseases. On the other hand, triaryl pyridines are identified as photosensitizer. They are extensively used in the phosphorescent organic light emitting diodes. Triphenylamine broadly used as an efficient whole transmitting material. That is why they are used as photosensitizer. Furthermore, pyridine analogs have a major role in supramolecules. These are some images of the same antim in which pyridine analogs are clearly shown. What we have strategies? Many strategies have been adopted so far for the synthesis of pyridine compounds. It is important to choose an efficient, feasible, environment-friendly, and economical approach to synthesize these compounds. Green chemistry has attracted a great attention of researchers from all over the world. It showed great results without any destruction of the environment. Actually, green chemistry offers various methodologies such as microwave irradiation, ionic liquids, ultrasound rays, supracritical fluid, solvent-free, and catalyst-free synthesis. Principle of green chemistry has a very vast field, prevent waste, maximize atom economy, synthesis of less hazardous material, harmless reaction conditions, non-toxic chemical products, use renewable feedback, avoid chemical derivatives, increase energy efficiency, use variety of catalysts, prevent pollution, design process for chemical degradation, minimize potential for accident. Solvent-free synthesis. The solid materials such as silica gel, alumina, and bentonite supported with acid are preferred catalyst. Catalyst performance in synthesis reaction results in a high yield product, least production of waste material, and no need of carbon chromatography in the presence of these catalysts. But we have started that we have prepared catalyst A, in which concentrated sulfuric acid is dispersed with a silica gel in dichloromethane and steered at room temperature for 4 hours. And similarly, we have used another catalyst which is concentrated acetic acid is dispersed in silica gel in dichloromethane and steered at room temperature for 4 hours. This is the pictorial work of catalyst preparation. In this slide, you can see that how catalyst is going to be prepared. This is the catalyst preparation process. After 4 hours, we got this catalyst which is used for our synthesis. Before going to use the catalyst, what we have did, we did the same analysis of the pure silica. and then that's diffused with acid. There is a clear difference between the morphology of the scene images of simple silica and fused with the acid. Then we optimize the conditions in which some physical characteristics of the catalyst, in which we have used the ratio of the silica with the acid, 1 into 0.25 and 1 to 0.5, 1 into 0.75, 1 to 1 ratio, and we have got different FTIR bands, and these by increasing the band of IR, it is clearly shown that our silica is diffused with the acid. The fused silica showed a variety of strong FTIR spectrum bands with asymmetric stretching vibration mode of the silica group with oxygen. Similarly, this is the catalyst B, in which similarly we have used the silica with the acetic acid in a different ratio, and every time we have got an increased band of FTIR spectrum, which clearly shows that silica is going to be dispersed with the acid catalyst. Reaction optimization was done with different catalysts. The best catalyst was selected in comparison with product yield and reaction efficiency in less time consumption. In this table, you can see that the 1:2.25 ratio and reaction time was 1 hour and 15 minutes and yield of 45%. Similarly, when we have seen that the best ratio was 1 into 0.5, in which we have got 55% yield in 1 hour. That's why we have selected this one, because otherwise the time consuming. This is the graphical representation in which by increasing the amount of catalyst, you can see the yield is increased. As the catalyst is increasing, you can see that product yield is increasing. This is the simplest procedure for the synthesis of pyridine derivatives, in which we have used different components. That's why we say that is the multi-component synthesis, and we have used this catalyst in the single flask, and we have obtained this tri-substituted pyridine. This is recyclable and non-corrosive catalyst, solvent-free synthesis, no need of column chromatography. This is the flow work in which we have obtained our product through successive steps in the lab. This is the mechanism through which pyridine derivatives have been synthesized. You can see in this slide that we have not used the pyridine anywhere. We are just going to synthesize the pyridine itself. Product 1 was obtained as brown steel gum in statistical yield. This is the picture of the compound, and its structure was confirmed with the FTIR and proton spectra, which show that there is some multiplates in the 7 to 8 region represents some aromatics in the compound. Similar compound 3 was obtained at 60% yield, and it was brown semi-solid gum, and its structure was confirmed with the help of FTIR and proton spectra. This is the product 4, in which 60% yield was obtained. It is sticky solid, and its structure was confirmed with the help of FTIR and proton spectra. All the synthesized derivatives are subjected to different biological activities such as antioxidant, antibacterial, and urease inhibition. Antioxidant controls the production of the free radicals that may lead to the chain of reaction and can damage the cells of the organism. Antibacterial to control the growth of various bacteria. Urease generation may cause peptic ulcer as process rheumatoid in the human being. All the synthesized derivatives were screened for anti-active potential were found to be inactive. The inactivation of this compound may be due to the flat geometry of the compounds. Interestingly, compounds have shown antibacterial activity when compared with the standard fraction, in which all the derivatives show proximal antibacterial activity. And urea inhibition, all the synthesized derivatives show potent activity. There is a 12.8 micromolar when compared with the standard drug thiourea. All the derivatives have potent activity of urease enzyme. Conclusion. In conclusion, 5 multifunctional pyridine derivatives synthesized by a highly efficient and economical method, the antibacterial, antioxidant, and urease inhibition studies was done. Interestingly, all compounds were found to be potent inhibitors of urease enzyme and also demonstrated good activity against various gram-positive and gram-negative bacterial agents. These synthesized compounds may serve as prime molecules in the recent drug discovery. Acknowledgement, this work was done by one of my MPhil students, Ms. Asha Praveen, and we have done in collaboration with Dr. Ina Makwa. She is Associate Professor at Lahore Women University. And I am grateful to my supervisor, Professor Dr. Mohammad Gal Chaudhary, because I did my PhD from this institute, and it's a great honor for me to be here after a long time. And I will pay my heartfelt thankful to my supervisor, mentor, Professor Mohammad Chaudhary, because he gave us some opportunities of doing the NMR spectrum and all the other analysis. And some HEC, I am also thankful to HEC because they have provided us fund under SRGP. The project was completed under SRGP grant. This is our university recently, because before it I was doing the job in the Lahore. After that I moved to the newly established university.
Any question, please?
Thank you for that very clear description of green chemistry and potential. One of the points of green chemistry is, of course, to make industrial process cleaner and less damaging. Yeah, and we just one of the points, one of the green chemistry is the potential to make industry cleaner in the production of core chemicals. You described it as economical, and if an industry is going to use green chemistry, they will always take economic factors into consideration. So this is a very difficult question. If you were going to scale it up to be an industrial process, is it cheaper or more expensive than the conventional way of producing viridine now?
Definitely, new methodologies are where take time and other cost is very high. But this is a laboratory work, but it can be transformed to the industry, but it will take time. Okay. Any other question?
Okay, thank you very much. Good. Next speaker will be Dr. Hannah Sanditi, Associate Professor of HEK, Research and Institute of Chemistry. I forgot, sorry, to give the Thank you for you here. Thank you very much. John, thank you. I'm sorry for that. I forgot. Dr. Khanna studied Associate Professor Engineer Research Council of Chemistry, Karachi University, Pictorial of the presentation, the small molecule graph conjugated, the respect of new biological activities. Dr. Sajidi has a 39 publication. She did PhD under the supervision of Professor Awad Chaudhary, and she has done 2 postdocs, one from America and another one from Germany. Thank you very much. If you are sorry. As you have a call, please don't talk loud.
Thank you so much. I'm habitual of making so many announcements, so feel like announcing again. Thank you, Chair, for a very generous introduction. And I want to start my talk with this disclaimer that I have sore throat. So kindly bear with me with fluctuation of my voice throat. So during the following 20 minutes or so, I'll be talking about a small molecular drug conjugate with the prospect of new biological activity with some of the examples of our recent work. So I want to start my presentation with a very basic question with organic chemistry and ask with the perspective of chemical university or university. Like how many organic molecules are possible on the face of the earth? Do we know the answer? Yes, of course, on the approximation basis or limited set of novel drug-like molecules in chemo, we can say that it is estimated that 10 raised to power 63 small molecular entities are possible. But and when I say small molecular entities, that means drug-like. And when I say drug-like, that means the compounds, the small molecules should follow the liquid scale, diversity score, and etc. And out of this 10 raised to power 63 molecule, only 10 raised to power 8 chemical entities have been synthesized, semi-synthesized, or isolated from natural sources. So this is actually a huge gap. So having said that, it is also important to know that only approximately 500 proteins have been targeted so far, and these are proteins of clinical importance. So this is the hydroprotein screening platform, and in order to target these 500 proteins and interrogate proteome and genome, it is important that we have new chemical entities, we have new chemical space. So there is a continuous space or continuous need to, for the influx of new chemical entities, because current drug development paradigm is still based on a screening of large number of libraries of compound. And it is therefore important to generate these libraries, and there are 2 possible approaches or method, you can say. One, high throughput natural product chemistry, or second could be combinatorial and parallel synthesis or medicinal chemistry. This diagram right here, A is chemical space where each dot is representing a chemical compound, small molecular entities which follow the lipid scale rule of 5. And these tiny dot, or maybe one of these tiny dot, you never know, is a life-saving drug. So we, as medicinal chemists, we work on learning cycle. What is learning cycle? This is basically nominating one of this tiny dot, which is actually a chemical compound, and then designing to synthesize this molecule eventually and test it. And if we are lucky enough, we need to repeat so many learning cycles, and if we are lucky enough to get the hit, we need to further optimize, and the cycle goes on until unless we find out this small little tiny dot right here. What is this? This is the ideal drug candidate. And when I say ideal drug candidate, that means it should have enough permeability, like it should pass blood-brain barrier, and it should have enough solubility, it should be selective, and metabolically stable, because there are lots of gut enzyme and etc. And it should have enough potency and synthetically accessible. You can easily synthesize and you can scale it up in the industry. So right at the interface, there is a drug candidate or drug lead. So this is the probing of the chemical space, which we medicinal chemists do. Like every other research, this probing the chemical space also starts with the idea that I mentioned, that you can get the idea from the chemical space in situ screening. biological active lead molecule can lead to some idea. And then based on this idea, you basically design chemical method to synthesize this type of molecule. And for library production, we generally go after 3 to 6 steps, not more than that, because for library, we need to make huge number of compounds, and we cannot afford to synthesize a chemical entity which actually will need many steps for the synthesis. So after that, you basically produce the library. And for library, you can use the facilitated protocols. Nowadays, so many advanced technologies are available. And then we conduct the biological screening, and if we get new hit, then the cycle is being repeated. Okay, there is some problem. Okay. So having said that, we also, in our report, we also wanted to increase the chemical space, and what we do for that, I really like this quote, Sir G.M. White, who's a Nobel Prize winner. He said, the most fruitful basis for the discovery of a new drug is to start with an old drug. And that's what exactly drug repurposing is. Drug repurposing is basically polypharmacological driven strategy, which basically is the adding value to the existing drug. Because, by targeting the other diseases other than the disease for which this molecule or the drug molecule was actually developed for. So this basically utilizes, the phenotypic screening, hydrophilic screening, and so on. Many drugs have been repurposed, and they were repurposed for other indications, like this methotrexate. It was originally a chemotropic agent. It was repurposed for immune system suppressant and many other. Like every approach, drug repurposing also has some limitation, because to get the new indication from the existing FDA-approved drug, the success rate is quite low. So to supplement the compound collection, the library of biological active compound, another approach is to start with the privileged structure. What are privileged structures? These are the pharmacophores with known biological activity. They are known to bind with certain target. So this is another approach that you can incorporate these privileged structure, and you can build your desired compound around these structure, and then you are sure to have certain type of biological activity, and you will be sure that these type of molecule will bind with certain target. So this is one way to enhance the rate of having selective molecule. So inspired with this, we were combining these all approaches in our work. And here I will give you just glimpses of some of our work. Okay, this is the one of our work which is published in Scientific Report. In this, we were able to, identify a dual inhibitor of urease and inflammatory marker. We just started with this drug isoniazid, which, as Salim also showed in his presentation, it is an anti-tuberculosis drug. By using simple chemical transformation, we were able to synthesize a library of small molecules, and then eventually we were able to identify these dual inhibitor. And you see, this isoniazid has this isonicotinol unit, and this pyridine type skeleton, and these are some of the FDA-approved drugs which have a similar skeleton, and these are some natural products with similar skeleton. So this is the simplest transformation, chemical transformation. Isoniazid would be reacted with isothiocyanate, and thiocyanate carbamazide library was synthesized. And this is the diversity handle, which created basically the diversity in our library. So quickly, in just one slide, these are the few inhibitors which we were able to identify. And you can see our compounds are far better active than, for example, this compound, than the standard acetohydroxylic acid. And these all compounds are, anti-inflammatory agent as well on the oxidative processing. And by the way, all these compounds were new, so we were able to publish in good journal. This is another example. We started with this sulfur methacine, which is basically antibacterial sulfur drug. and it is known to suppress the synthesis of dihydrofolic acid. So we were able to synthesize a library by diversifying this handle with the various variety of acyl halides and this type of amide moieties or amide compounds were synthesized. And again, we were able to have a very potent anti-inflammatory agent. All of our compounds were actually tested for various inflammatory markers, including PMNS, nitric oxide radical scavenger assay was performed, and TNF alpha was also determined, and many of our compounds were, better active than the standard. So these are some of the members of the library. So in this slide, you can see the comparison. All the reds are basically better active than the standard compounds. The good news is that all of these compounds are non-cytotoxic in the normal saline assay. This is another example. In this case, we utilize this hydrochlorothiazide, which is a basically diuretic drug, and it is being used for the treatment of high blood pressure. What we did was we introduced a triazole moiety within the skeleton of this drug, and inspiration obviously was obtained from lots of existing drugs like ribavirin. It has triazole. This drug, and this is an antifungal drug again. It is triazole containing drug, and this one as well. So what we did was simple click chemistry. And this is the main scaffold or main drug. We propaginated it, and then we obtained these two products. We were largely interested in this single propaginated one, and then we couple it with pre-prepared azide, and azide were prepared with benzyl bromides by the reaction of sodium azide, well-known reaction. And then with this, triazole moiety was introduced, and the library was produced, and all the compounds were purified by column chromatography and then characterized by usual NMR mass and IRUV and other techniques. And many of these compounds are new, and we were able to identify many good inhibitors of alpha-glucosidase enzyme. Alpha-glucosidase, everyone knows that it is the enzyme which is responsible for maintaining the increasing the blood sugar level. So if we can inhibit the alpha-glucosidase, indirectly we can control the hyperglycemia. So we were able to have very potent compounds, and each one of them is better active than the standard A-carbos. And we were able to file a patent, local patent based on these compounds as well. This is another example. We actually synthesized the ester conjugates of ribavirin. Ribavirin is an antiviral drug. So this is the structure of the drug, and we were able to synthesize many derivatives out of it. So this is the reaction scheme which we followed. We started with this ribavirin, and then we made this ketal right here, and this ketal was eventually reacted with chloroacetic acid, and then this ether was actually synthesized, and then this was followed by the further esterification at this unit. And then 33 compound library was synthesized, and these are the member of the library right here. All of these compounds are new, not reported before, and we were able to test them against SARS-CoV-2 activity. What we did was we conducted a plaque reduction assay, and we identified that out of these 33 molecules, this compound, this window, can we remove this window from here? We were able to identify this compound L was actually very potent inhibitor of COVID SARS-CoV-2, almost comparable with the remdesivir. And as a proof of hypothesis, we wanted to check, the activity of various intermediate involved. So what we did was we went back and we synthesized all the intermediate involved again, and then we tested each of the individual intermediate against same assay. And then we found out that this intermediate A is also having very good activity, but not as good as compound. And in addition, we also tested the reagent as well in order to check whether the activity was because of the reagent or the compound which we have synthesized. So we were also able to determine the point of treatment point or mode of action of these compounds, and we these kind of studies are still in process, and but we were able to identify that our compounds are possessing therapeutic and prophylactic effect. So this is the ongoing work. This is another example. In this we were we wanted to design and synthesize the fatty acid and other conjugates of tenofovir. This is the anti-HIV drug. What we did was we conduct lots of chemical transformation, multi-step chemical synthesis, and we were able to synthesize a library of 120 fatty acid and other conjugate based on this tenofovir skeleton. So, just to give one reaction scheme, we started with this alanine, we protected it, and then we obtained this protected amino acid, and which was further reacted with variety of alcohol, and then it was again deprotected and was made reacted with the tenofovir, and then these photosubstituted phosphoramide derivative were synthesized, and which eventually were coupled with various phenols. And in the last step, the introduction of various fatty acids were introduced, and we were able to synthesize these fatty acid conjugates. All of member of these library were actually tested against a variety of biological activities, including anti-inflammatory, anti-Leishmanial, and anti-HIV, and many more. So we were able to identify lots of good anti-inflammatory agent against reactive oxygen species. The main basic screening assay was performed, and we see many of our compound, actually all of these compounds are better active than the standard ibuprofen, and all of these compounds are non-cytotoxic as well against normal 3T3 cells. See these compounds, they are all better active. I'll just quickly pass by, so that we can save time. All of these compounds, these are like weak to moderate active. And we also tested our compounds against breast cancer cell lines, and we found out that some of the compound, like compound number 32, 150, and this 148, are very good active, comparable with the standard doxorubicin. So further studies against these compounds are on their way. So we are studying these compounds for further secondary assays or not. These are some other members of the library with relatively lesser activity, but still comparable. These are some of the results for the anti-Leishmanian activity. We were able to identify many good inhibitors of leishmaniasis. For example, these compounds are not as good as amphotericin B, but certainly comparable with pentamidine or multiphosphate. So these are some other members of the library I quickly pass by. In conclusion, medicinal chemistry continues to play a major role in drug research and development with the help of new approaches and techniques in drug repositioning, characterization, privilege structure, redressing has emerged as an alternative approach for the traditional drug discovery. And based on our work, we were able to enrich the chemical space with new chemical entities with diverse biological activities. These are some of the US and local patents we were able to file based on this study. In the acknowledgement, I would like to acknowledge that first I am the grantee of ISNPC twice, and the project funded by CERN and MTD Network. And these are my collaborators. This is my small tiny research group, and very focused group of student has worked on this project. This I would like to thank you all for your kind attention. Thank you.
For your beautiful lecture.
Thank you.
Any question for this?
Yes.
Thank you very much. It is very nice presentation. I can ask about the today's design. Let's say use a purple for the positive control.
Then oxidase,
Like
alpha glucosidase. Yeah, we generally use a carbose, but that has quite high IC50.
Yeah. At the same time, working by a different mechanism, by similarity to the polysaccharides which are consumed for the body.
I am not sure about that.
I mean, maybe the mechanism is different.
Yeah, maybe, but generally we use this one for.
Thank you very much.
Thank you.
You? Okay. Thank you, dear.
Thanks.
All the foreign delegates requested get together at the reception of guest house before 6:30. Vehicle will move 6:30 from guest house. And we thankful for the presence in today's session. Also tomorrow morning at 9 a.m. session will. Thank you.